Our body has two main types of genes that control cancer. Oncogenes, which drive the formation and the growth of cancer, and the tumor suppressor genes, which work to stop it. In previous videos, I talked about the HER2 as an example of an oncogene and the BRCA as a tumor suppressor. In this video, I will introduce another tumor suppressor, p53. In fact, p53 is the most well-known tumor suppressor linked to many different cancers, and it has drawn enormous scientific attention ever since its discovery. p53 was first discovered in 1979, but figuring out what it actually did was so challenging that it took scientists nearly 10 years to confirm that p53 is a tumor suppressor. Once that discovery was made, interest in p53 exploded. In fact, in 1993, Science magazine even named p53 the molecule of the year. This was the cover of Science at that time, and here's the article itself. The title, p53 Sweeps Through Cancer Research, really shows just how intense the excitement around the p53 was back then. Moreover, in 2017, the journal Nature published an article titled The Most Popular Genes in the Human Genome. In that article, the author looked at which genes receive the most attention not only in cancer research, but across all areas of the life sciences. And as you can see in this chart, p53 was the clear leader. This shows that p53 has continued to attract enormous scientific interest even more than 40 years after its discovery. So, what exactly is p53 and why is it so important? In normal cells, p53 works as a powerful tumor suppressor. But p53 is one of the genes most prone to mutation. When p53 mutates, it loses its ability to protect the cell. And in some cases, the p53 gene can even be completely deleted. Whether through the mutation or deletion, the loss of a p53 function removes key safeguards, making it much easier for cancer to develop. So, how often does a cancer involve the loss of a p53 function? According to a well-known study from 1994, more than half of all human cancers have either p53 mutation or a loss of a p53 expression. In other words, in at least 1 out of every 2 cancer patients, p53 is not working properly, which shows just how important this gene is. So what are the differences between cancer patients with and without the p53 mutations? Many studies have shown that cancers with p53 mutations often lead to worse treatment outcomes. For example, a study published in 2022 followed 68 people with a triple-negative breast cancer. All patients received chemotherapy first to shrink the tumor, and then underwent surgery. After surgery, 25 patients had no remaining cancer, while 43 still had residual disease. Among these residual tumors, some had a normal p53, while others carried p53 mutations. The researchers then monitored these two groups to see whether their cancer came back and whether they survived. This shows the percentage of patients who remained recurrence-free over time. The blue line represents the tumors with a normal p53, and the red line represents the tumors with a mutated p53. As you can see, recurrence-free survival declined much faster in the mutation group. For example, 3 years after surgery, 77.8% of patients with a normal p53 were still recurrence-free, compared with only 56% of those with p53 mutations. This clearly demonstrates that triple-negative breast cancer patients with p53 mutations have a worse prognosis than those with a normal p53. Then why is the loss of a p53 function associated with the poor prognosis? We often use terms like cancer growing, recurring, or metastasizing. When we say cancer is growing, it means that the number of cancer cells is increasing through cell division, reflecting their strong proliferative ability. Recurrence means that some cancer cells survive the treatment, demonstrating their high survival capacity. And the metastasis refers to cancer cells moving to other parts of the body, reflecting their strong ability to migrate. Importantly, p53 can influence all these three behaviors. So how does it do that? Before we answer this question, let's first take a look at how regulation works. To regulate something, you need two opposing forces. For example, to control a car's speed, you need both the accelerator to go faster and the brake to slow down. Cells work the same way. Regulating cell growth requires signals that promote growth and the signals that suppress it. In this system, p53 functions as a brake. It slows down or stops cell growth when necessary. So if p53 loses its function due to mutation or deletion, cells can no longer stop growing, and thus keep dividing without control, just like a car with broken brakes can't stop. Likewise, cell survival is controlled by a balance between signals that promote survival and the signals that trigger cell death. In this system, p53 also works as a brake. It promotes cell death. So when p53 malfunctions, cells gain a much stronger ability to survive. This is why cancer cells lacking p53 function often withstand anti-cancer treatment and eventually recur. Cell migration is also regulated by signals that promote or inhibit movement. And the p53 again acts as a brake. So when p53 is lost, cells become more mobile, making them more likely to metastasize. Overall, p53 suppresses cell growth and migration and promotes cell death. So when cancer cells lose p53 function, they gain advantages in growth, movement, and survival. This is why cancers with p53 loss tend to have a worse prognosis. They are more likely to recur and metastasize. Therefore, restoring p53 function in cancer cells could be a promising strategy for cancer treatment. Several approaches are currently being studied, and I will introduce them in a future video. Thank you for watching.